get across here. But there's a lot of other interesting things you can do if you can control the cells and their environment in the length scales of the cell. So this thing here is probably like, again, length scale of your diameter of your hair. That small. So you can kind of take that, and this is the same aggregate of stem cells. So what happens in the body, if you have an kind of developing stem cell aggregate, is that it's very organized, right? Your kind of different tissues, your heart always comes up on the left hand, there's lots of organization. But what happens if you do this outside the body is you lose all that organization. So you don't have no longer the kind of these stem cells differentiating in a controlled way. But what you can do with these kind of techniques that I mentioned is that you can start recreating that. So here we take the same stem cell aggregates that I've been talking about, and we put them in two different environments. And this, so the general idea is what a cell sees determines what a cell does. So a cell that sees one environment starts behaving differently than a cell in the same aggregate that sees a different environment. So, and this is what happens. So you see this aggregate is placed at the interface between these two different materials. And as you look at it over time, this is as from initial to day 7, you see that on one side there's a lot of things happening, and on the other side it's not. And this is, again, because of this control that you can have on these stem cells. You can start taking these aggregates and polarizing them and try to have different parts do different things and actually direct the differentiation cells in different ways. So that's another very interesting thing. So the other things that I want to talk about, I want to kind of move a little bit quicker because I want to talk about some of our tissue engineering work. So let's say we do all this stuff that we did for stem cells, and we can actually make cells that you can use for different applications. There's still a big difference between a bunch of cells and a functional tissue. So how do we bridge this gap? So one of the ways that we try to do this is to try to engineer this tissues with controlled architecture. So I mentioned that most tissues are very complex in their organization. So if you look at your muscle, you see a lot of fibers. This lobule structures I mentioned. This is your kidney. So you actually see this is the functional unit of a kidney. It's a channel with a lot of cells doing different things. And then your pancreas generates islets, and these islets actually secrete insulin that regulates glucose. So what we want to do is to try to recreate these kinds of structures and do it at the same length scales as these tissues, as the microstructures of these tissues. So one of our approaches is to try to say, can we actually engineer a functional unit using kind of engineering approaches that I'm going to talk about, and then be able to assemble these functional units together. What's interesting about each of these functional units of the tissue that you see here is that each one of the units has all the metabolic or mechanical function of the tissue. So the fiber, for example here, the myofiber, can actually contract. And the way your muscle actually functions is that you have a lot of these fibers packed together to actually to make the larger tissue. So we wanted to kind of use this principle to make small building blocks and then assemble them together to get the larger tissue made. So we've been working with a lot of different types of materials. I don't want to talk too much about material science here, but just to kind of get one term across. So we work with hydrogels, and hydrogels are kind of what they sound like: hydro, water, and gel, right? So I kind of like to think about them as like, if you see them, they're like Jell-O, right? All of you guys have seen Jell-O. So we work with Jell-O-like materials. And so there's a lot of different types of hydrogels, and we play around with all these different ones. And once we have these Jell-O-like materials, what's the characteristics of Jell-O? There's a lot of water in Jell-O, right? And in that way, Jell-O is similar to your body, because your body also has a lot of water in it. So what we try to do is we try to take these Jell-O-like materials and actually start putting cells in them and use different kind of engineering approaches to control the shape. So here we can fabricate them in the shape of these little blocks. We can take different kinds of materials that are found inside the biological system, so like same kind of chemistry that exists in your body. We can take it, take those kind of materials and make these, put cells in them and make these little building blocks. So there's a lot of interesting ways we can do this. We can use this as a molding process. You literally put the cells and the jello before it's actually cured. It's still liquid. We can put the cells and this jello thing. We can mold it and get these little building blocks. And then there's lots of other ways you can do it inside little fluidic channels and a variety of other things. So I'm not going to talk about all the different ways you can make these little jello building blocks. But the idea is, with these jello building blocks, these are again, you can kind of think about these as the same thickness of your hair. Very, very small stuff. So we can take these very little small stuff that have cells inside the jello, and we can now start to say, how do we take these and start assembling them together to get more large, complex stuff that has the same complexity as your natural tissues do. So, this is some examples of how these cells behave inside these jello-like structures. So here, this is a big gel. And this is cells initially. You see that the cells are rounded. And after a little while, in these kind of gel-like materials, they start spreading inside, and they kind of start communicating with each other and then generating something like a random structure. But in other cases, in this case where you have this jello-like material actually have an architecture, in this case a fiber-like architecture, then you start seeing the cells actually sense the architecture. So this is a fiber of a gel. And the cells sense the architecture and start aligning uniformly in the direction of this fiber. So the cells actually sense the fiber and align and elongate and differentiate along, which is much more controlled than this system. So this is the kind of things that the architecture of these small scale is actually important. So we can take these building blocks, which I mentioned, and the idea is, can we actually start putting them together in a very organized way to generate the complexity of tissues. So, so we do this using a number of different approaches, which I just want to kind of give you the glimpse of this. So some of these approaches involve very simple things, right? So if you have little cubes of water-like material and you want to assemble them, then each one of you guys knows about oil and water don't like each other. So you can use similar kind of thing. If you put a drop of water in oil, it all kind of comes and makes this little spherical droplet. But now if you put these little small cubes of very water-like materials, then they start assembling. So you can actually, so if you put water or a lot of excess water, you get these droplets. But as you put these little gels and you don't have too much excess water, then you can get them packed in a very controlled ways. So, and they're very biologically compatible. So this green basically means cells are okay. So you can see that once they're packed together, the cells are okay. And there's other things that one can do to add more control to this process of this assembling of these little gels. You can, for example, have these lock and key kind of structures, so they assemble together in controlled ways. So these rods assemble around these little kind of crosses, and lots of other stuff. So I don't want to talk about all the different ways you can induce the assembly here. But needless to say, like, one of the things that people are very excited about in my area is how we can try to assemble these things and get tissue-like structure. So it's someone who's very, someone who's interested, we can talk about it more. And what we're doing now with these kind of structures, these little packed gels, is we're actually trying to now put them in the body and see whether we can recreate the function of the tissues that we're interested. So we're actually have some work in both bone and heart cell reconstruction to try to use these approaches in the body. So in the last 5 minutes, and I promise not more than 5 minutes, I want to talk about another very interesting thing to try to add to generate tissues. So what I talked about so far doesn't take you to consider the vascularization, your blood vessels. So we've also been very interested in making blood vessel-like structures by using these kind of microfabrication systems, or the same kind of things as your kind of cell phone chips, same kind of technologies. So what we do is, instead of